Our technology is called single-chain trimers of peptide MHC complexes. Basically, in the past, when people have tried to make these peptide MHC complexes, they relied on bacterial expression protocols that require the individual expression of each of the two subunits of the MHC. In other words, expressing the HLA subunit individually and then the beta-2M unit individually as well. After that, they have to synthesize the peptide or the antigen, as shown here in red, and then they have to refold all three components together in a in vitro setting. The limitations behind this process is that when it comes to scaling up to generate maybe hundreds of these reagents, it is extremely laborious and time-consuming. And so our advantage with the single-chain trimer technology is that we can encode all three components of this protein into one single genetic construct on a plasmid. And we do this with the assistance of these amino acid linkers that link the antigen to the beta-2M and then the beta-2M to the HLA. And in doing so, we have complete control in a genetic manner over the identity of the antigen that we introduce and over the identity of the HLA allele that we use to make these peptide MHCs.
What does this enable for us to do?
Well, I think our paper demonstrated the power of this technology primarily in the context of SARS-CoV-2, just to show that this was an effective technology for a pandemic, that we could rapidly assess antigens in this manner and then also identify TCRs. But you can imagine that the SARS-CoV-2 is but one of many potential different infections one can have. This also includes bacterial infections, fungal infections, and antigens might also be found in autoimmune disease and also, as you said, Jim, in cancer. So I would say that this technology offers a huge advantage for the immunology field in terms of identifying these antigens from two different standpoints, from a standpoint of scale and a standpoint of speed. In terms of scale, we've shown in this paper that we can now look at thousands of antigens. And for something like cancer, especially, that would be very important because patients can have anywhere from tens to thousands of different mutations, depending on what cancer types they present with. And then the second advantage is speed. So we're now that we're able to generate potentially thousands of these proteins within a small time span, maybe less than 1 or 2 months, this enables us to capture a patient population that has a much more severe onset of symptoms. So in cancer, for example, the prognosis might generally be very poor compared to COVID. So that's where I think this technology has a huge advantage.